Exercise number 1B, Interphase, Replication of Genetic Material.
Hello everyone! Today we will talk about the importance and the structure of DNA. DNA is made up of monomers which we call nucleotides. These are molecules composed of a deoxyribose sugar with a phosphate and a base attached to it. These nucleotides are attached to each other in strands via phosphodiester bands to form a sugar-phosphate backbone. The band formed is between the third carbon atom and the deoxyribose sugar of one nucleotide known as a 3' and the fifth carbon atom of another sugar on the next nucleotide known as a 5'. So there are two strands running in opposite or anti-parallel direction to each other. These are attached to each other throughout the length of strand to the bases on each nucleotide. There are 4 different bases associated with DNA: the cytosine, guanine, adenine, and thymine. In normal DNA strands, cytosine binds to guanine, and adenine binds to thymine. These two strands together form a double helix. So now, we will talk about DNA replication and why this process is important. But before that, let me introduce you to the cell cycle. A cell cycle is a series of events that takes place in a cell as it grows and divides. A cell spends most of its time in what is called interphase, and during this time, it grows, replicates its chromosomes, and prepares for cell division. The cell then leaves interphase, undergoes mitosis, and completes its division. The resulting cells, known as daughter cells, each enter their own interphase and begin a new round of cell cycle. Cell cycle is the name we give the process through which cells replicate and make 2 new cells. Cell cycle has different stages: G1, S, G2, and M. G1 is the stage where the cell is preparing to divide. To do this, it then moves into the S phase, where the cell copies all the DNA. So S stands for DNA synthesis. After the DNA is copied, and there's a complete extra set of all the genetic material, the cell moves into the G2 stage, where it organizes and condenses the genetic material, or starts to condense the genetic material, and prepare to divide. The next stage is M. M stands for mitosis. This is where the cell actually partitions the 2 copies of the genetic material into the 2 daughter cells. After M phase completes, cell division occurs and 2 cells are left, and the cell cycle can begin again. DNA replication, also known as semi-conservative replication, is the process by which DNA is essentially doubled. It is an important process that takes place within the dividing cell. Why replicate DNA? DNA is the genetic material that defines every cell. Before a cell duplicates and is divided into new daughter cells through either mitosis or meiosis, biomolecules and organelles must be copied to be distributed among the cells. DNA found within the nucleus must be replicated in order to ensure that each new cells receive the correct number of chromosomes. The process of DNA duplication is called DNA replication. Replication follows several steps that involves multiple proteins called replication enzymes and RNA. In eukaryotic cells such as animal cells and plant cells, DNA replication occurs in the S phase of interphase during the cell cycle. The process of DNA replication is vital for cell growth, repair, and reproduction in organisms.
DNA replication can be thought of in 3 stages: initiation, elongation, and termination.
In initiation stage, DNA synthesis is initiated at particular points within the DNA strand known as origins, which are specific coding regions. These origins are targeted by initiator proteins, which go on to recruit more proteins that help aid the replication process, forming a replication complex around the DNA origin. There are multiple origin sites, and when the replication of DNA begins, these sites are referred to as replication forks. Within the replication complex is the enzyme DNA helicase, which unwinds the double helix and exposes each of the two strands so that they can be used as a template for replication. It does this by hydrolyzing the ATP used to form the bonds between the nucleobases, therefore breaking the bond holding the two strands together. DNA primase is another enzyme that is important in DNA replication. It synthesizes a small RNA primer which acts as a kickstarter for DNA polymerase. DNA polymerase is the enzyme that is ultimately responsible for the creation and expansion of the new strands of DNA. In elongation stage, once the DNA polymerase has attached to the original unzipped two strands of DNA, the template strand, it is able to start synthesizing the new DNA to match the templates. It is essential to note that DNA polymerase is only able to extend the primer by adding free nucleotides to the 3' end. One of the templates is read in a 3' to 5' direction, which means that the new strand will be formed in a 5' to 3' direction. This newly formed strand is referred to as the leading strand. Along this strand, DNA primase only needs to synthesize an RNA primer once at the beginning to initiate DNA polymerase. This is because DNA polymerase is able to extend the new DNA strand by reading the template to 3' to 5' synthesizing in a 5' to 3' direction as noted above. However, the other template strand, the lagging strand, is antiparallel and is therefore read in a 5' to 3' direction. Continuous DNA synthesis, as in the leading strand, would need to be in the 3' to 5' direction, which is impossible as we cannot add bases to the 5' end. Instead, as the helix unwinds, RNA primers are added to the newly exposed bases on the lagging strand, and DNA synthesis occurs in fragments but still in 5' to 3' direction as before. These fragments are known as Okazaki fragments. In termination stage, the process of expanding the new DNA strands continues until there is either no more DNA template left to replicate at the end of the chromosome, or two replication forks meet and subsequently terminate. The meeting of two replication forks is not regulated and happens randomly along the course of the chromosome. Once DNA synthesis has finished, it is important that the newly synthesized strands are bound and stabilized. With regards to the lagging strands, two enzymes are needed to achieve this: the RNase H and DNA ligase. RNase H removes the RNA primer that is at the beginning of each Okazaki fragment, and DNA ligase joins fragments together to create one complete strand.